Today we will be talking about amino acids acting as acids, bases, and buffers, okay? Because sometimes, well, most of the time actually, amino acids can deprotonate, and they can act as bases, or they can act as acids, right? And so this whole video is going to be focusing on isoelectric points. And essentially, towards the end of the video, you should be able to calculate the pi. So you should be able to calculate this guy right here, which is the isoelectric point. So it's essentially when an amino acid is electrically neutral, and you're going to find out the pH at neutral. Okay? So, what does that mean for us? We have this little table right here, and essentially towards the left, you're going to see that these pKa values correspond to different amino acids. So the pK1 is essentially the COOH portion, okay? That's essentially the COOH, and right here would have to be the NH3, the NH3 right here, okay? 3 plus. And the pKr is the pK value for the R chain. So, for example, in tyrosine, you have the amino acid, and let's see if you can actually draw it out, right? So for tyrosine, let's do that in blue, classic blue. Tyrosine, it would be this right here. We have NH3 plus. We're going to have COOH, and then you will have this guy right here, CH2. Okay, so this whole red chain, well, specifically this portion right here, that's going to have a pKr value of, let's see, 10.9. Okay, 10.9. So that's the table that you can have as we do these practice problems, okay? So I'm not sure if the professor is going to give you a pK table during exams or whatever, or he's just going to ask you to memorize like one or two of them. I don't know. In general, sometimes you can get away with using a pK value of 2.2. That seems to be the average. Sometimes it's not going to work. For instance, cysteine has a pK value of 1.8, right? Or threonine, or threonine has a pK value of 2.6. So sometimes it's not going to fly, right? pK2 value, it's like really just out there. The minimum being 8.8 and the maximum being 10.8. So it's really ridiculous to do that. Okay, so now we're going to talk about the different points of the amino acid. Whenever you're finding out the pi value, you always want to start really, really acidic. So what do I mean by that? I mean that you want the amino acid to have all the available protons in it, okay? So for instance, this is kind of like the general format for the amino acid, right? So you have your amine group, you have your carboxyl group, okay? But notice that they gave an extra proton to start with. So typically, it's usually like this, right? Right, so you're used to seeing that. But whenever you're first starting off, you want to give the carboxyl group a proton, because you want the amino acid to really act as a base. You want to have enough protons to give away during the reaction, okay? Because we're going to be focusing on different points of the reaction, right? So right here we have we're going to be giving away some protons, right? So what happens if I add some base? Well, if I add a base, this hydrogen is going to go away and it's going to connect to the base. Okay, it's going to connect to the base, and it's going to give us a negative, COO negative, okay? Now, the reason why we start off with carboxyl, right, so the reason why we take off the hydrogen from this guy right here is because it's more acidic. It's actually more acidic than the amine group, okay? So remember that on the table right here, let's just use alanine, for example. Just use that as an example, okay? So let's say that this is alanine. So we have a CH3 right there. And this pK value is going to be 2.3, okay? And the amine group is going to be 9.7, okay? Cool, 9.7. So first of all, since it has a lower pK value, it's going to give off the hydrogen first. So that H bonds with the base, okay? So now that is going to be negative, and you still have a pK value of 9.7 for the amine group, okay? For the amine group, this guy, right? Now, if I add more base, okay? Let's say I get it to like 10. So I add more base, so I put at pH of 10. Let's say that this was 5, right? Then the base is going to overwhelm the amine, and the amine is going to shoot off a hydrogen. Therefore, it's going to lose a hydrogen and become neutral. Okay, that's fine. So to recap, if you enter a location where the pH is greater than the lowest pH, in this case 2.3, you will shoot off a hydrogen and become more negative, okay? So if we enter a location where the pH is 10, then the pH of 9.7 for the amine is going to shoot off a hydrogen. The reason why we can't shoot off a hydrogen for this guy right here is because he doesn't have one, okay? He doesn't have one. So now we had to figure out the electronically neutral structure. So what does that mean? You just want to have a Zwitter ion. So a Zwitter ion is just a neutral amino acid. Okay, so remember when I was talking about amino acids, I said, hey, you know, there's an acid and a base here. Well, if you have a negative on the amine, okay, and then you have a positive on the, so if you have a negative on the carboxyl and then a positive on the amine, what does that make? It makes it neutral, because a positive plus a negative is 0. So +1 and -1, those go up to 0. Okay? So here we go. Right here, we have a overall charge of the plus, right? So that is cationic, right? And if you remember the plus, you can think of the little cat, right? So that's the cationic structure, right? I think it's really cute. Alright, so if we lose a hydrogen right here on the carboxyl section, you're going to become neutral, because that's a positive and that's a negative, and together they make a neutral. And that's what we want. We want to find the PI value for that, okay? And over here, you have nothing and a negative. So overall, that is a negative. So that is the anionic form, okay? An anionic form. Okay, so how do we find the PI value? Well, the PI value, or the isoelectric value, is actually equal to the pK1 plus the pK2 divided by 2. Okay, so you take the average. So, you know, for instance, you don't want to take 3 values and divide it by 3. That's a no-go. So for instance, if you're doing tyrosine, you're not going to put 2.2 plus 9.1 plus 10.9 divided by 3. No, you don't want to do that. You seriously don't. You just want to have 2 values. And those 2 values are going to be from the left and the right side of the neutral substance, okay? So we're going to get into that later. I'm going to do an example for tyrosine, most likely. But in this case, the value over here is going to be 2.3. So we start with the really acidic value first, okay? So 2.3 for the carboxyl group. And over here was 9.7. Okay, I'm not using 5 or 10. No, I'm using the actual pK value for the groups. So the pK value for the most acidic portion right here would be the carboxyl group of 2.3. And then I'm going to use the most basic value over here, which would be for the amine, 9.7. Okay, so for the pi of, let's say, alanine, that's going to be 2.3 plus 9.7 divided by 2, which gives me a value of 12 divided by 2, and I think, I don't know, I'm just guessing here, I think that's 6, right? So that's 6. What does that tell us? It tells us that at the pH of 6, okay, you are going to have a alanine amino acid that is electrically neutral, okay? It's electrically neutral. So the reason why you should actually know why amino acids are electrically neutral is because, well, you know, sometimes whenever you're working with like protein structures, you want to, how should I say this? So the reason why it's important that you should know the isoelectric points is because amino acids react differently whenever they're exposed to either an acidic environment or a basic environment. And that's important because amino acids make up protein structures. Depending on if an amino acid is too acidic, that protein structure can change. So you might have different amino acids doing different things in different parts of the body. And it becomes really complex, and it really comes down to when you're linking amino acids together. You know, sometimes you won't be able to connect valine to leucine because they're in different forms. Okay, so it really depends on both the protein structure and the polymerization, so like the linking of amino acids. You can't put two things that are like completely different pHs together, okay? You want to have like a range, so like 6, 6.1, 6.5, you know? So amino acids that are closely linked together by pH would be ideal, okay? So that's why you need to know about pi values. So here is a example using tyrosine. Okay, so this is tyrosine. So whenever you write an amino acid, always make sure to write out the name and the abbreviations, okay? So there we go. It's actually Y. I keep forgetting. So it's tyrosine, right? So here's the initial reaction, okay? So whenever you're working with the amino acids, you always want to have an excess amount of protons. So you give everything the added protons, okay? So that means we have H3N+, we also have COOH, okay? We have COOH. And now looking at the value over here, tyrosine, this value, or I should say this baby right here, this bad boy, he's going to have a value of about 2.3. So let's do that. So he's going to have a value of about, let's see, 2.2 actually. Okay, so I'm looking at this column. Okay, and now if we enter a zone, let's just make up a pH, I don't know, like 2.3. Okay, then the H is going to shoot out and make this what? Negative. Okay, so now we have a positive and a negative. So that must mean that the overall charge is what? It's 0. So positive and a negative is 0. So we have a charge of 0. Over here we had a positive and nothing else. So the overall charge would be positive. Okay. Now if we enter a zone, let's say 9.2, then we're going to shoot off a hydrogen from the amine. Okay, because this guy right here has a value of 9.1. Again, looking at this chart right there, okay? So because it's in a basic environment, you're going to lose a hydrogen. You become H2N. This is still negative, and this is going to be neutral, okay? Actually, yeah, so this is going to be negative, and I don't know why, but I'm missing an H right here, so that's not good, right? That's not good. We're going to put this as an H right there, and this is going to be negative, right? So overall, this charge right here is negative. So the overall charge is negative 1, okay? Negative 1. So now what happens if we enter a value that is more basic, okay? So what if we enter here? So let's make this right here. And I hope you're having a great day, of course, obviously. Okay, so what if we enter a value of about 11, okay? If we enter an environment where the pH is 11, well, right here, the oxygen of this guy right here, he's going to release his hydrogen. He's going to release his hydrogen. He's going to be acting as an acid, okay? And that makes it negative. This part is also negative. So overall, you have a net charge of negative 2. Well, that's not really good, is it? That's not good. Because you want the amino acid to be electrically neutral, okay? You want it to be electrically neutral. So you pick the compound that has a net value of 0, okay? Which is this guy right there. And then, to find the PI, you need to have the value, okay? So the PI of this would actually be the what? 2.2 plus the 9.1 divided by 2, okay? Because there are two values. Now the reason why we didn't pick the value of 10.9 is because that's not going to give us an electrically neutral compound. Notice that this value right here and this value right here, okay, are actually very close to the electrically neutral amino acid. Alright? So to make it more convenient for you, I'll just change these values. Let's say that directly at 2.2 and 9.1 you're going to have a reaction, okay? So let's just say that to make it make sense. Therefore, the electrically neutral amino acid is between those two values, 2.2 and 9.1. Between those two values, somewhere in that pH range, you're going to get an electrically neutral amino acid. And what do we get whenever we have that? Let's see, 2.2 plus 9.1 divided by 2. So 2.2 plus 9.1 is 11.3. And then we're going to divide that by 2. And so we have the PI value of about 5.65. Therefore, at the pH of 5.65, you should be expecting a neutral tyrosine amino acid, okay? So therefore, you now know how to calculate the PI values for amino acids. If you like graphs more, if you're a visual person, I understand. Well, let's say that this is for alanine, okay? Let's do a titration, because most amino acids, in fact, all of them actually, can be titratable. So what is titration? Titration is just when you add a little itsy-bitsy baby amount of acid or a base to a solution, okay, in order to either decrease or increase the pH. So let's say that we always initially start at the most acidic portion of the graph, or at the reaction, right? So whenever you have so much acid, everything has protons. So for instance, this carboxyl group has the proton, right, the H portion, okay? And we start titrating. We start adding some bases and bases and bases. And eventually the pH gradually increases. It gradually increases, okay? So now you get to a point where you still have the H for the amine, but you no longer have the proton for the carboxyl group, okay? And this is the isoelectric point. So if you take in calculus or advanced mathematics, usually the inflection point for the curve, the little flat part of the curve, where it's like almost 90 degrees, is the isoelectric point, okay? So that's the isoelectric point, which means it's neutral, okay? And if you keep adding bases, then you go from the neutral point and you keep going and going and going until you hit a certain area where you no longer have the proton on the NH3. you actually have NH2, and you no longer have the proton because that was gone a long time ago. And if you keep adding bases, then eventually you'll just exponentially increase in basicity. So if you notice something, there's a flat part right here, right? And then there's a flat part right here. And essentially that region is called the buffer capacity. What? It's like everything I say, builds up on each other. How wacky and weird. It's not like I planned to do that or something. But that is the buffer capacity, okay? So the buffer capacity is essentially the maximum or minimum amount that the buffer can kind of like efficiently stop the exponential increase of acidity or basicity, all right? So once you get to like a region right here, let's say over here, if you add any more acids, you're just gonna go all the way to like 0 very, very quickly. If you add some bases right here, then you're gonna go to like the pH of 14 very, very quickly if you add like a millidrop of bases, okay? So always the isoelectric point is between the buffer capacity, okay? And of course the buffer capacity, let's call that BC, is equal to plus or minus 1 of the pK value, okay? So it's the pK value. Always remember that. Oh, and remember something else, that at the inflection point, or at the isoelectric point right here, so in this example for alanine, 6 pH, right? The pKa is going to be equal to the pH. So whenever alanine is at the pH of 6, you can always say, hey, since the alanine is neutral, it has a pH of 6, that must mean that the pKa is also 6, okay? It's also 6. For tyrosine, we said that it was 5.65. Well, that must mean that the pKa is also 5.65. In fact, the pKa is always going to be equal to the pH whenever you have a mixture of half acids and half bases, okay? So that's called ionization. I don't really like that term. Just imagine if you had a box of donuts, okay? And personally, I like custard and raspberry filled. So if I have a dozen donuts and 6 of them are custard filled and 6 of them are raspberry filled, then we can say that the pKa, or the custard filled, is equal to the amount of the raspberry filled, or the pH, okay? So the acid is equal to the bases, therefore the pKa is equal to the pH, right? And that is literally the only location where the pKa is equal to the pH. That's very important for you to know and for you to remember on exams. So hopefully this made sense to you and helped you out to find the isoelectric points, okay? Of course. Hopefully you write out these structures and try to find the isoelectric point for like 5 of them, I don't know. And hopefully you have a great day and that this video helped you. Thank you so much for watching and for spending time with me. I really appreciate you, and I know that you can get through this course, right? You're smart and you work hard. Remember that I love you and that I appreciate you. Thank you so much and have a great day.